So I mentioned in the last chapter how much technological development actually changes science. So it's a continuous cycle of scientific advancements lead to technological advancements, which then lead to scientific advancements, and so on and so forth. So technology has been incredibly instrumental in changing the way that biologists work. So again, everything is really based on different measurements. So it's the determination of the dimensions of something using a standard unit, which the units we talked about in Chapter 0. Again, we also talked about accuracy, a description of how close a measurement is to the true value of the quantity measured. And then precision is the exactness of a measurement, which is taken over several trials. Okay, so again, you have this here where you have precision when they're all grouped together like they are in these two targets here. And then you have accuracy when your hits are basically grouped around the bullseye. Okay, and this basically these values kind of average out to be within the bullseye. Okay, so another term that you need to be aware of is a microscope. There are several different types of microscopes. The one that you're most likely familiar with, or the most familiar with, is known as a light microscope. Microscopes are just devices that provide an enlarged image of an object. So they help you look at things that are really small, hence micro. So different examples of microscopes. You do have a light microscope here, which is shortened as an LM. So this is looking at the stoma in a leaf. Okay, so these are the little openings on the bottom side of leaves that let oxygen out and carbon dioxide in. So we'll talk about these more later, okay, when we start talking about plant biology. So this is a light microscope. With light microscopes, the object can't be incredibly small. The light microscope just doesn't have that fine precision to get down to really, really small detail. It also cannot be living, okay, for the most part. So you will actually have to take like slices of things. Unless they're like microscopic organisms, then they can be living. But for the most part, especially like in this example here, you would have to do a very thin slice of the leaf in order to be able to see this. Okay, because the light does have to pass through the sample in order to see it. You also have, in this case, what's known as a scanning electron microscope, or an SEM. So this actually gives you a lot more detail about kind of the topography of whatever you're looking at. So here you can see the stoma is actually slightly raised from the rest of the leaf. Okay. And then you also have what's known as a transmission electron microscope, or a tunneling electron microscope, or TEM. So this gives you a lot more kind of cellular detail. Okay, so you can see the stoma is actually the gap that is between these two, what are known as vacuoles, that hold a lot of water in them. And so there is like this just little gap that appears between these two vacuoles, between these two guard cells is what the technical term is actually. So you've got a lot more cellular detail here. Okay, there is no topography detail like there was with the SEM. You also can't necessarily see into the cell itself like you could with the light microscope, but it's still, basically with microscopes you generally don't look at any one type of microscope in isolation. Oftentimes you will use multiple different types of microscopes to get information about the same cell, okay, or the same sample. So we do have this video here about the history of the microscope, so make sure that you watch that. So some more technology that has increased the way biologists function, okay, biologist research. Now we're getting into medical. Okay, so not microscopes, but now we've got ways of looking inside the body. So first up, you've got X-ray images. So X-ray is actually hits you with slightly like with X-rays. So there's a lot of energy involved going through that. The X-rays will actually pass through your skin and through any soft tissue in your body, and then they actually will get stopped by any hard tissue or excessive liquids. Okay, so that's why you have bones will show up brightly on X-rays. Okay, you also have MRIs, or magnetic resonance imaging here. Okay, so this is showing you not just the bone, but also some of the soft tissue surrounding the bone here. Okay, so here you have a very good definition of the femur. Okay, so the bone in the thigh, and then the tibia and fibula are here. That's the bone in the shin. And then you can also see your patella, so your kneecap here. Okay, and you can see some of the soft tissue and the tendons and everything that connect all of these bones together and make them work together. You also have what's known as a functional MRI. So this is looking at a brain scan here. This is basically taking an MRI, but it's looking at function instead of just what's there. So there's a lot more that goes into an fMRI, oftentimes done with brain scans to check that everything in the brain is actually functioning correctly. So complex systems with all of this and research and everything, complex systems are oftentimes modeled on computers. So computer models can be used to study systems that cannot be studied directly. So you see this oftentimes with proteins. You can't actually look at the protein itself. It's too small for that. So instead, using X-ray crystallography and some different techniques, scientists can create these computer models where you can actually go in and click and drag the protein around so that you can see the shape of the protein and where everything kind of connects. So they will also use these complex systems. Sorry, my dog is itchy, apparently. But they will use these complex systems to model things like heart attacks. So when you have a normal heartbeat, okay, you'll see regions of contraction and relaxation within the heart, okay? And they are supposed to be in very specific regions. They are very contained, and that's keeping the rhythm of the actual normal heartbeat. During a heart attack, all of that normal rhythm gets thrown off. So you see contractions and relaxations in very different patterns. Okay, so that's what they're tracking in these two images here. So you see the normal heartbeat with its very distinct patterns of contractions, and then with the heart attack, you can see it just looks crazy. Okay. You can also track the effects of medicines on the human body. So this is, oftentimes you will see kind of depictions of the blood flow carrying the medications around the body and then where it gets affected, and tracks basically the effect of the medications on different organs and all of this kind of stuff. You'll also see movement of water molecules into and out of a cell. This is actually a very common thing. You might think that cells try to hoard water, but they really don't. There is a lot of movement into and out of the cell with the water because the water oftentimes has various nutrients or waste products in it, dissolved in it. So the cell either wants to take them in if it has nutrients in it, or it wants to get rid of them if it's kind of washing out the waste. You'll also see computer models with the spread of disease through a population. So oftentimes scientists, when there is any kind of epidemic or disease spread, they try to trace back to what they call patient zero. Patient zero is the first one to come into a specific area with the disease. And so they're the ones who are basically responsible for passing it on to everyone else in that area. So, I mean, they are very good at tracing back to patient zero, especially with certain diseases like HIV. They actually were able to trace back to patient zero over several decades, which was pretty amazing. But and that's how we know that HIV actually originally was the simian or monkey version of the disease. And then patient zero apparently ate a monkey brain, a raw monkey brain, because that is considered a delicacy in certain regions of the world. And the SIV, the simian immunodeficiency virus, actually mutated to survive in its new host, and thus HIV was born. So, computer models are used when experiments are either not safe, ethical, or practical to do on the actual subject. Okay, so you're not going to basically rip someone's heart out just to see the contractions in a heart attack. Okay, because I mean, that would be weird, first off. Secondly, you would kill the person. And third, once you disconnect the heart from the rest of the body, you're not going to get very many contractions. So, that kind of thing. Alright, so you do have, well, some of the tools that are kind of revolutionizing biology have actually given rise to new biological studies. So you have the tools of molecular genetics. This is dealing with DNA and then how DNA is actually used inside the cells to create the function of the cells. So a gene is actually a segment of DNA that stores that genetic information. So it's a gene for a particular trait. Okay, so you might have a gene for, say, blue eyes. Okay, actually you have several, but that's another chapter. These tools gave rise to the study of genetics. So genetics is actually, at least the way it is now, it is a relatively new science. I actually have my minor in genetics, and it's studying really the DNA, the genes of the populations, which leads us straight into population genetics, which is why genetics was not my major. It was just my minor, because I hate population genetics, but we'll talk about that later in the year as well. You also have biochemistry came out of this. So biochem is looking more at, alright, so you've got these genes, you've got the DNA, so now let's take a look at how they get, or how they create these proteins, which then go on to do the functions in the cell. So this is looking at your genes here. So this is just showing 3 genes, A, B, C, and D. So through our understanding of DNA, we have actually been able to study genetics on a molecular level. So we can get down and manipulate DNA on that molecular level. So that is molecular genetics, and that's how you get things like GMOs. So we have actually input frost-resistant genes into some orange trees in Florida, which means that a late cold snap is less of an issue for them. Okay, their orange trees are better able to survive that kind of thing happening because of this manipulation of DNA. A lot of this is not necessarily in food, but it's in research. So like you can manipulate bacterial DNA to insert genes. And then genomics is the study and comparison of genomes both within and across species. So oftentimes you'll find very similar genes in different species when you wouldn't necessarily think you would. So we'll get more into this as well, but if you ever see crime scene shows and they talk about DNA fingerprints, they are basically referring to molecular genetics. Okay. All right, so that is the end of 1.3.